Biology Part 74. At the same time, the chemical contents of the platelet granules are released, causing more platelets to become attached to the plug and promoting further vasoconstriction. The platelet plug forms quickly, but it is mechanically weak and is replaced by a more robust blood clot, as described in the next section. Number 4, blood clotting. The most important steps in the blood clotting are summarized in our figure today. A clot is formed when the soluble plasma protein fibrinogen is converted into insoluble threads of fibrin. These threads form a mesh which traps the formed elements of the blood and changes it from a free-flowing liquid into a solid gel. The formation of fiber is catalyzed by the enzymes such as thrombin. This is not normally present in the blood but appears after vessel damage. As you can see from the diagram, the thrombin production does not occur directly but follows a chain and a cascade of linkage reaction. These involve plasma protein clotting factors, most of which are manufactured by the liver. At each step, the inactive form of an enzyme is converted to an active form, which catalyzes the next step in the chain. Some of the enzymes require Ca2 ions as cofactor. Some of the enzymes require Ca2 ions as cofactors. Others require phospholipids, which are released from the platelets as they accumulate to form a plug. Vitamin K, as they form to a plug. Well, vitamin K does not function as a cofactor but is needed in the synthesis of prothrombin and certain underclotting factors. Within the body, clotting is normally confined to the immediate vicinity of the vessel damage. This localization is possible because of the blood plasma and the tissue contain additional enzymes which inactivate the various clotting factors almost as soon as they are formed. Contrary to popular belief, exposure to air has nothing whatever to do with blood clotting. Number 5, clot retraction. Well, once formed, the blood clots are reduced in size by a process called retraction. This action depends on the platelets, which send out cytoplasm projection, and pseudopondia, which adhere to the fibrin threads. The cytoplasm of the platelets contains contractile protein strands which then contract, pulling the threads and making it there, promoting an efficient final repair of the damaged vessel. This enzyme is produced there, pulling the threads cluster together to form a stronger, denser clot. Fibrin is eventually broken down and the enzyme plastomin with that enzyme is produced from its inactive form of plasminogen. When clotting first takes place and acts very slowly within the clot, progressively dissolving the fibrin and promoting an efficient final repair of the damaged vessel. Diseases associated with hemostasis include various kinds of hemophilia in which blood falls to clot. The condition occurs in the absence of any one of many essential protein clotting factors normally present in blood plasma. The commonest type is caused by sex-linked recessive gene as explained earlier.